What I would expect is we're going to see sex-specific interventions in aging and aging-related diseases. Even diseases common to the male and the female, like Parkinson's and Alzheimer's, that having an intervention that's tailored to the male or the female will be more efficacious.
From the USC Leonard Davis School of Gerontology, this is Lessons in Lifespan Health, a podcast about the science and scientists improving how we live and age. I'm Professor George Shannon, Kevin Chu Chair in Gerontology. On today's episode, how Professor John Tower is advancing what we know about the role of sex differences and the role of mitochondria in aging. John Tower is a professor of biology and gerontology. Findings from his lab at USC Dornsife are uncovering pathways for how lifespan may be extended and revealing why some interventions have opposite effects on males and females. He recently spoke to us about his research. So, what does your lab focus on?
Well, we're focused on basic mechanisms of aging, and we're using the fruit fly Drosophila melanogaster as our experimental model because of its very powerful genetics and its very convenient model organism to work with. And we're particularly focused on the role of sexual differentiation and the role of the mitochondria in aging. So as you may know, in humans, women live longer than men, and the reason for that is not entirely understood. And also malfunction of the mitochondria, which is also called the powerhouse of the cell, is directly implicated in aging and multiple aging-related diseases, including Parkinson's disease and Alzheimer's disease and cancer. And so we'd like to understand at a very basic level, why does the mitochondria malfunction during aging? And does this or does this not have is this related to or a result of sexual differentiation of the male and female?
So, antagonistic pleiotropy is a long-held hypothesis of aging, first proposed by the biologist George Williams. What is that, and how does your work add to it or alter our understanding of it?
Well, there's no consensus in the aging field on pretty much anything, but I would say at this point, antagonistic pleiotropy is the most favored model for how the genetics of aging works across species. And the idea is that genes can be beneficial in one context, but detrimental in another context. Specifically, they're likely to be beneficial early in life, promoting things like differentiation and growth and sexual reproduction. And in the long term, the same genes are detrimental and have a cost during aging. Probably the first gene that was pointed to as being a candidate for exhibiting antagonistic pleiotropy is the famous tumor suppressor gene P53. In young animals, including young humans, it's a very potent tumor suppressor. It's critical for preventing cancer because it sends cells with DNA damage either into the apoptotic pathway where they self-destruct or the cellular senescence pathway where they're stopped from dividing. In either way, that is a strong prevention of cancer. However, during the aging of the animal, some of the cells in our body that are supposed to keep replicating, like our stem cells that replenish our blood and the lining of our gut, they can also accumulate enough DNA damage or telomere erosion that they will go into the apoptotic or senescence pathway, and then that's then detrimental for us being able to maintain our tissues.
So you've talked about antagonistic pleiotropy. What is sexual antagonistic pleiotropy?
Well, I think I started out making an assumption that was not well-founded, and that was if I wanted to study basic mechanisms of aging, I assumed that they would be the same in males and females, and it wouldn't make much difference which sex I pursued for the experiments. And we used males because they were more convenient for technical reasons. The females lay eggs, which would tend to make the food sticky and create a hazard for the flies. So if we used males, we could avoid that. But about halfway through our research, we were looking at the gene P53, which is conserved between humans and Drosophila. And what we found was that when we made mutations in P53 or altered its expression, it did affect lifespan, but it did it differently in males and females. And that made us really re-examine our assumptions about aging in males and females when such a basic gene with basic conserved functions, why would it affect males and females differently? And so that's when we started looking at sex differences in aging and sex differences in the function of genes during aging. And in the course of that, I've made a complete about face from thinking that sexual differentiation was not important to thinking that, well, maybe sexual differentiation is actually causative to a large part in the aging process. In other words, in differentiating the male and the female, you set up the situation for sex-specific trade-offs between reproduction and aging. And some aspects of these trade-offs are common between the male and the female, and some of them are unique to either the male or the female, in that there are pathways that promote reproduction, but then have a cost for the long-term maintenance of the animal. That's the kind of antagonistic pleiotropy my lab is focusing on right now, which is the idea that a gene can be beneficial to one sex, but detrimental to the other, or a gene could be detrimental to each sex in different ways. One of these genes we've identified, we didn't identify this gene, but we've studied it. It was identified by other researchers. It's expressed in the male, and it's called sex peptide. And when the male and the female mate, the male introduces this sex peptide hormone into the female. And this hormone hijacks the female's physiology, makes the female make more eggs, but it also reduces her immune function and reduces her lifespan. And you might ask, why would such a gene evolve? And the thinking is, well, this is beneficial to the male because the female can store sperm after she mates for about a week and a half. But then she could also mate again with another male. So the thinking is that natural selection has acted on the male to create this gene that essentially forces the female to make more eggs with his sperm at the cost of her resources that she might use for a subsequent mating. So he's forcing her to make offspring with his sperm as opposed to having her save some of her resources to make offspring with some future mate.
That is truly fascinating. And it doesn't take a genius to understand the application to human beings as well.
There are some intriguing parallels in humans. For example, imprinting in male and female mammals. The male imprints genes that result or favor the growth of the fetus and the extraembryonic tissues, whereas the female is imprinting genes that tend to reduce the growth of the fetus and the extraembryonic tissues. And this has also been interpreted as natural selection acting in the male to try and force the mother to use more of her resources for his offspring, and natural selection acting in the female to try and limit how much resources she puts into that first offspring so she can save some for future offspring.
So picking up from that, why are the X chromosome and mitochondrial genome significant for aging and longevity research?
Well, there's several reasons. The X chromosome, which is in 2 copies in females and 1 copy in males, that's common for both humans and Drosophila. And that means that the female has 2 copies of every X-linked gene and the male only has 1. And there's increasing evidence that the female takes advantage of that extra copy to yield extra activity of those gene functions to the benefit for stress responses and ultimately for longevity. With regard to the mitochondria, now that is one of the features of biology that has been shown to be quite conserved across species during aging. So across species, we see a decrease in mitochondrial gene expression and mitochondrial gene function during aging. And the relevance to sex is that the mitochondria is transmitted to offspring only through the mother. And so this means natural selection can only optimize mitochondrial gene function for the female. This means that the male inherits a mitochondria that is less optimal for his physiology than it might be. And so what we see is that mitochondria isolated from female mammal tissues function better than mitochondria isolated from males, consistent with this hypothesis. And so this may be one reason why females tend to live longer than males.
In my classes, I'm always trying to explain that by approaching it from a social context, since that's what I know about. So this is really informative and useful for me as well. We usually associate game theory and the prisoner's dilemma with social or computer science, but they've been used in the context of describing the relationship between a cell's nucleus and mitochondria. How does this apply to or inform your work?
Well, our idea was that the observation is that mitochondria are shorter lived than the nucleus. So if you look in the cell, there's typically one nucleus, multiple mitochondria, and the mitochondria are always being degraded and remade. So the mitochondria is inherently shorter lived than the nucleus. And we hypothesized that this might be a strategy that the mitochondria uses to make the cell dependent on continually making new mitochondria. So a strategy for its maintenance that allows it to be maintained and to be maintained as a separate entity from the nucleus.
So when it comes to genes, then aging may not always be a negative trait. Can you explain that?
At the level of the organism, we think aging is always a detrimental trait. But when you look inside the organism at genes and macromolecules, you see that there are sometimes situations where it's beneficial for a gene or a macromolecule to be short-lived. And so that instability, if we want to think of that as an example of aging, means that being short-lived is actually beneficial in the context of collaborating genes.
What you just said about aging not always being a negative trait, that it does seem to exist within cells, doesn't it? Cells, if they don't die, can become cancerous.
The inherent instability of cells in terms of the fact that they're engineered to undergo apoptosis if they are missing the appropriate signals or if they've gone through too many cell divisions, that indeed is clearly an anti-cancer mechanism. And so you can think of this inherent instability of the cell as beneficial in one context, as in that it helps prevent cancer in the young animal. But the same mechanisms that are engineered into the cell to promote its self-destruction in response to DNA damage or telomere erosion, in the long term during aging, that function may become detrimental.
Mm-hmm. Could this also be relevant to gene editing, like the Nobel Prize-winning CRISPR?
Well, yes, in as much that those groundbreaking tools for editing genes are the tools that we and others would use to test these hypotheses. It makes it more possible to alter genes' functions as well as their potential stability in the context of this model and actually test these models.
Speaking of models, how does computer modeling inform your work?
Well, we've used computer modeling to test the idea that one gene being more unstable than its partner could be beneficial. And it's computer modeling that's given us the first indications that indeed that can be true, that when two genes collaborate in a replicator, in a structure that will replicate itself, that it indeed can be beneficial for one gene to be less stable than the other gene.
On our podcast, we've heard from researchers who use particular worms or fish to study aging. And your studies have focused on a fruit fly. Why is that a good model for studying aging?
Well, there's several reasons. The majority of the genes in the fruit fly are conserved with humans, including the majority of human disease genes are conserved in the fly, as well as the majority of basic biological processes. So it's very highly related in many ways to humans. Also, it's very convenient for aging studies. It's got a fast life cycle. It only lives a couple months, and they're relatively cheap and easy to culture in the laboratory.
Mm. What did you discover in your recent work with the drug mifepristone in fruit flies that point to the possibility of extending human life?
It was a surprise to us. We found that this drug mifepristone, which is in humans a glucocorticoid and progesterone receptor antagonist, if you fed it to mated female flies, it dramatically increases their lifespan. And that's a surprise because those targets aren't thought to exist in the fly. So we're not exactly sure what its direct target is, but the lifespan increase is profound. And we've also, in collaboration with Sean Curran's lab, found that it can also increase the lifespan of mated C. elegans worms. So the fact that it has this lifespan extending effect in mated females in flies and worms, which are as distant from each other in evolution as either one of them are from humans, made me think it's possible that it could also have a beneficial effect in humans.
So do these findings apply to both males and females?
So far, we've only seen a benefit for lifespan in females.
Well, that's really interesting. What future directions do you hope to explore in this research?
We're trying to get down to the real mechanisms for why the fly dies and how these interventions are preventing death. We're not there yet. What we're looking at in the near future are trade-offs between reproductive metabolism and the maintenance of the somatic tissues. So our results hint at the idea that in the mated fly, there's an increased absorption of amino acids from the gut for energy and to make lipids, and that the fly is shortchanging the turnover of muscle and other somatic tissues. And so by normal maintenance of tissues requires that they're always being turned over. And if you were to shut that down, you would expect those tissues to start losing their function. And so that's what we want to test specifically in the lab next.
What is the pattern for translating your research ultimately to human beings?
Well, we're trying to develop quick and easy assays in the fly for things like the turnover of somatic tissues and the responsive lifespan to drugs, using flies that we've engineered to be fluorescent so that we can measure these events happening in the fly without killing the fly. So you can assay the fly at multiple time points, you can assay large numbers of flies, and what we're hoping that will facilitate is screening of drugs, that we'll be able to screen for drugs and small molecules that will increase the lifespan of the fly and decrease these aging-related changes and allow us to screen through quite a few chemicals in hopes of finding ones that might be relevant for humans. Well, I think what I would expect is we're going to see sex-specific interventions in aging and aging-related diseases. Even diseases common to the male and the female, like Parkinson's and Alzheimer's, that having an intervention that's tailored to the male or the female will be more efficacious.
Is there anything that we haven't touched on that you feel might be important just to finish the discussion?
Well, one other aspect we're pursuing is the role of the microbiome. Some of our research also suggests that when the fly mates, it becomes more susceptible to the toxic effects of its own microbiome. And we're going to try and test that hypothesis specifically by making germ-free flies and adding different bacteria back one at a time, or at least that's our future goal.
Well, thank you very much. I'm really shocked that in all the time that I've been at this school, this is the first time we've really had a chance to talk. So I, for one, am really pleased to have this discussion. Thank you so much.
Well, thank you. It's been fun to do.
That wraps up this lesson in Lifespan Health. Thanks to Professor John Tower for his time and expertise, and to all of you for choosing to listen. Join us next time for another lesson in Lifespan Health, and please subscribe to our podcast at lifespanhealth.usc.edu. Lessons in Lifespan Health is supported by the Ney Center for Healthspan Science and the Center for Lifespan Health.